Welcome back. So this afternoon we have Paul who's going to tell us about modeling using Cryo-EM data. Thank you, Paul.
Thank you for the invitation. Yeah, as you can see, this is an overview, as far as I understand it, of the problems of modeling into cryo-EM reconstructions and the software that we have to hand for addressing those problems. We can further refine that is to mean model building, refinement, links, and ligands. And towards the end of the presentation, I will be segueing into how one might use Coot for those operations. So I'm going to divide the sorts of problems that we face overall into these 5 sections. What's this blob? I have a reconstruction, and I don't know what I'm looking at. I have some expectation of what this reconstruction should be, this piece of protein or this domain or this complex, what this complex is, and I expect that there's a particular sequence there. Let's say that we've docked a structure into the reconstruction, the rotation translation, and now we need to make it fit. Then we have the details of the side chains and the ligands, the pose, refinement, and issues arising with dealing with the ligand and the interaction of the ligand with the protein, for example. So let's look at the first one of those. What's this blob? Oh, my intention, by the way, is to finish in 55 minutes past the hour. 50 minutes plus the 5 minutes that we delayed. Okay, so and then you'll see towards the end of the presentation that this is, there's lots of figures and screenshots towards the end. It's relatively lightweight. We may have time for a demonstration at the end if you're interested in that. Okay, so with that out of the way, so one of the first of these problem domains is, what's this blob? You have a cryo-EM reconstruction and a sequence, or a set of sequences. And to address this, there are automated methods. And in the CSP4, we have Buccaneer, written by Kevin Cowton. And more recently, there's Modelangelo from here, Pathwalker from Stephen Lutker and co-workers, and there's also Phoenix Auto Build. There are references here. If you want this, I'm happy to share the PDF with you if you wish. Secondly, we might want to say we have found a blob, or part of the design of that, the way we could couch that problem is we have a piece of chain that we could trace, and you can use various programs to identify what that protein is. So imagine that you have some part of the reconstruction you think you didn't expect it to be there, but it's there. What is it? There is Check My Sequence from ARP-WARP, or a spin-off from ARP-WARP. What I'll be talking about for a couple of slides is Coot's attempt to address this question. And as you may know, I'm sure you do know, in fact, that Modelangelo can address this question as well, and that's it's in the publication process at the moment. In the problem domain, the first example of this that I saw discussed was Cryo-ID from Zhu from UCLA, and his then postdoc who moved to Columbia, I think, who described Cryo-ID. That is to say, they took malaria parasite and did some vague, rough purification, and then had some reconstructions on the grid. They didn't know what it was, but they knew what the species was. if that is the situation, then how can we go about identifying what the protein is? So that's the example here. We have a reconstruction. We know the set of sequences. We know the genome. Can we identify what the protein is? So this is from the paper from Greg, and this is from the paper from Kiresh, which is under review. Right, so how does Coot do it? So the idea is that we have some built fragment. You can do a fit helix here and then do add terminal residues rather readily with little brain to create a 40 residue fragment. And then you feed that fragment, that polyalanine fragment, into the Coot identified protein, and every side chain position, you give it the map, of course. Coot identified protein, which is a separate program, compares a hypothetical rotamer at that position with the likelihood distribution of that rotamer type, and it's been trained on all proteins better than 4 angstrom in the PDB. So it has some probability distribution for those side chain positions given that side chain orientation. And then we scan across for each residue type, each rotamer type of each residue type, and then compare the density that we're actually looking at a side chain position with the probability distribution, or the likelihood distribution, that we've learned from an analysis of the structures in the EMDB. It looks like this in the output. It can see, this is a structure with which I was familiar from a few years ago. Laurie Passmore and colleagues, the cleavage and polyadenylation factor. Anyway, the protein is not that important. I just built into a piece of the structure here, and it has identified a sequence, and it's given it a score. What I've done in the next slide is plotted those scores, so the log likelihood, or minus log likelihood, versus the null hypothesis. The null hypothesis is just a Gaussian at the C beta position. And unfortunately, these numbers are negative, which was disappointing. But at the same time, it was able to identify the correct solution. You can see that the best probability, the highest probability, is the one that was the right solution. So we were able to identify this protein, the CPF, from the whole of the genome of the yeast. This is a protein of picture that I showed, different protein. This is a parathyroid hormone receptor. This is actually the parathyroid hormone that I shared with my colleagues a few months ago. It's both an indication of progress and also failure. Because what we're doing is putting the program in guess the sequence mode, and you can see that it's found some correctly, identified the side chain correctly out of 20 amino acid types, but it doesn't like to find tryptophan. Well, I've seen lasting. You could forgive it for that, but it does like to put tryptophans in. For some reason that I haven't yet identified, and I'd like to get back to this at some stage, but as you can see later on, I've been a bit distracted with the GUI instead of the scientific part of Coot. And so another part of the questions we could be asking about our reconstruction is, I have a protein model and a reconstruction, where is my domain in that map? So this is like the molecular replacement question we might be forming if we had X-ray data. So I think it's useful to call it in Cryo-EM molecular placement. The tool that we have been using, myself and my colleagues have been using here is MolRep, but there are other tools available, Phoenix, Docking Map, and EM placement. More on that in a moment. There are also some other tools, as Ron mentioned the other day, Citus in particular by Willy Riggers, and that's had an update since 1999, but it's a long-term player in the field. Okay, so for MolRep then, written by Alexei Vagin, it is written in Fortran in an old style, has an awkward and confusing interface. It was, however, the basis of our sequenceless molecular replacement that we wrote up, or Alan in particular wrote up in 2015. Alan Brown, previously of this neighbourhood. Also mentioned by Ron yesterday, the day before, a few days ago. What we're going to do, or this is the way we make it work in Coot these days, is to extract a fragment from the map, which is to say, you can say, well, here's a blob of density. I don't know what it is, or don't know the orientation, but I have some expectation that a particular domain is there, and then Fourier transform is used on the map to calculate structure factors, and the problem is treated like molecular replacement. That was written up in that paper. And so this was an example of it working using the Balbez database. So there was a blob of density. We went through the map looking to say, we had a prior model from the bacteria E. coli, and there were some extensions, a considerable number of extensions to the reconstruction that were as yet identified. We said, okay, so here's a blob. And then there were 20 other similar blobs, not exactly similar, but 20 other examples, and they were tested using the same protocol. This is the best success. The Balbez database was used to identify the protein without knowing anything about the sequence, and then having the correct solution in terms of rotation translation. The rest of that protein was identified and built. So the extraction from a fragment of the map is now built as a Coot extension, and it runs in 5 or 10 seconds, something like that, depends on the size of your domain. But something like 20 or 30 angstrom radius is about 10 seconds. The sequenceless molecular replacement tool that was written up by Alan has undergone bit rot, and now no longer works on our computers. They used to be using the Sungridge engine, and now they will need to be resurrected if we're going to do that to work with Slurm. And instead of using Balbez, the protocol would be, I imagine, to be using AlphaFold instead to get a better or more accurate set of starting models for whatever it is you may have in your reconstruction. Right, phaser-EM placement needs half maps, and it turns out that only a minority of recently deposited structures, so I did a quick scan over the last year or two, only a minority have half maps, unfortunately, and this was recently 2023, that should be, not 13. I asked Randy for his slides, and he sent me over a few, so I won't dwell on the details here, but I'll leave them in the PDF. So the idea, as I have said, is how can we place an atomic model, a component, into a map? It's a 6-dimensional search. We have rotations and translations. And how do we go about scoring our solutions? And of course, as you know, phaser is entirely couched in terms of likelihood and log likelihood gain, as they call it. And in particular, what makes phaser particularly powerful is its ability to have an expectation of what a good scoring solution should be. How do we know what the final solution is? Right, so there are cases where this sort of tool is useful, and that is when the size of the domain is not the same size as your model that you're trying to fit into it. So it is by just, you can't fit it just by looking. So that's the sort of problem. That is perhaps some small fraction of cases, but it's not zero. So as you know, phasers use molecular replacement, a rotation search, and then a translation search. And the tools or the algorithms that have been used for that can be re-implemented or reused in the implementation of this tool for Cryo-EM. So it's a 6-dimensional search, as I said, break it into rotation search and translation search. And the idea is that you have some expectation of what you would get as a score based on a null hypothesis, which Randy's calling an uninformed model, which I guess is just a big ball of atoms. Okay, the expected log likelihood gain is the score that you can use to distinguish the best scoring model from the correct solution, or to identify the best scoring model as the best solution. Okay. So this has been added into or made available as a tool for Chimera X, and I let you know that it exists, but I haven't used it myself. Coot has the jiggle fit solution also, if you don't want to use MolRep, if you want to use pure Coot. But at the top of this, there's also a solution called fit in map in Chimera X. You can fit a model to a map or a map to another map. Global search does as Coot does, starts with a set of initial models in random orientation. Okay, so where does I mentioned AlphaFold 2. Where does AlphaFold, or how does AlphaFold or its successors fit into our world? Well, primarily for us, it's going to be used as a better source of initial models. We had been using Balbez, and that's great, but AlphaFold 2 models, AlphaFold 3 or who knows what, or derivatives thereof, will be more accurate than that. One of the things AlphaFold 2 did well at is obviously at the CAST, the Critical Assessment of Structure Prediction, but that scoring system doesn't target long-range distances, and that was not a target of the people who devised AlphaFold 2. So that's not something that they're going to get right when we place their models in our reconstructions, which is to say the domain-domain juxtaposition and positioning will need to be adjusted, let's say. Additionally, if you only go in with the sequence, there's no, hey, what's this blob? What's this strange thing? I can't identify it. Issue that occurs when you get experimental data. That looks funny. There's none of that in AlphaFold 2 models, and there is routinely. What is that strange thing that I'm looking at in experimental data? It's not us who will be most influenced by AlphaFold 2. It seems to me that the people who will be most influenced are those who want to be using structural data, or now can do their science in the light of structural data without needing experimental data, and that is a group who's rather larger than experimental structural biology. Okay, moving on. Which map should we be using for refinement? There's a tool in Phoenix for that, Phoenix Auto Sharpen. And Phoenix Resolve Cryo-EM is an algorithm that takes 2 half maps and does density optimization, or solvent flattening, that sort of thing. So Coot has a wrapper for RefMac, and you can use that to generate a MTZ file, which you can then read in, and on reading that you can resample the map. And additionally, there's a built-in sample resampler in Coot as well. So this is a case that several people in the last couple of years have discovered, 5A1A, where the map was rather sharper than perhaps it needed to be. And you can see there's a particularly unfortunate, perhaps, a piece of structure where we have a cis peptide in front of a residue that's not a proline. That's unusual, and particularly in this case where there's no little to no experimental evidence to support that particular configuration. So what I'm going to do is run a set of, or display a set of reconstructions that have B factors applied. And you can see by adjusting the B factor, you can see that there is indeed a better place that we can move around that peptide or that few residues there to better fit the map. And so it's a good idea, generally speaking, to be looking at an array, a series of, it seems to me, this is what I do, at least I recommend it to you, a series of differently sharpened or blurred reconstructions that helps your interpretation of the map. Right, moving on to considerations of refinement. So Rob Nichols and Gerib some time ago worked on this idea where one could use previously solved template structures to provide structural information that helps stabilize low-resolution refinement. And it is the restraint generation part of ProSmart that we will be considering. So the idea is that we have a previously solved holo form with the sialic acid in the binding site, high-resolution structure. And this week, or this month, sometime later, we have this lower resolution data set where there is no ligand and it's low resolution. What we want to do is to stabilize the refinement of this protein given structural information from the high resolution structure. And the way to do that is local distance restraints. If I compare the two structures, can you see there that there's some opening perhaps of the domain? Maybe you can see that. So what we do is take the high resolution structure and ask of each atom, what are your neighbors? What's the distance to those neighbors? And then we can apply those distances to today's model. And those distances may be recapitulated, they may be longer, or they may be shorter. And that's what we're looking at here in these colors. So red doesn't mean wrong. Red means longer. And that's not unexpected when you have a ligand missing from a ligand binding pocket. You expect the domains to open up a bit. So most of the contributions to the target functions from the various geometric criteria in Coot use least squares or the Hooke's law, which is to say, if I double the distance of a particular atom along away from where I expect it to be, at say 1.54, to pick a number at random, and I move it away from that to 1.5, then I expect to pay a penalty. And if I double that distance, then I expect to pay 4 times the penalty. Now, when we're modeling proteins, that sort of target function turns out to be not the best. And we can use this modifier here, or M estimator, first promoted by Gemmon and McClure, to tamp down the contributions to the target function of this restraint when we're a long way away from the correct solution. So it cares when it's close, and then when you get so far away, it says, okay, I give up. I don't care anymore. Any change you make along this axis when you're out here is not going to change the blue line, so it doesn't care. Okay. So in Coot, so ProSmart allows you to give a previous or pre-calculated model, previously derived high-resolution model. In Coot, we can use the similar sort of thinking, but using self-restraints, which is to say that the starting model is the reference structure, and the distances can be used from that, so they're all exact to begin with, and then we try to fit it to the density and they get distorted to some extent. Keep the local environment similar to how they are when you started, but try to fit to the density. And we can change the weight to allow the fit to density or preserve the structure, and I'll show you how to do that if we've got time to see an example. So unlike Coot, RefMac has an alternative method, which is based on the second order, and it changes this. Sorry, it changes the second derivative, and that's called so-called jelly body stabilization. It canonicalizes the shifts in a way that results out to be similar to pro-smart-like restraints on local distance restraints, but functionally they're calculated differently. There's a program provided by Fei Long called libg that allows not only protein restraints but also stacking and pairing restraints using canonical Watson-Crick based pairing, and now more recently with wobble and reverse wobble bases too. So how does one use Coot? This is an example of structure from Jude Short. What we've done here is refine the structure without restraints in the first case, and then use the reference structure. And you can see that the structure that's built into the density and the reference structure don't agree well. You can see that they're moved apart. So that when we turn on the refinement with these restraints in place, they become more canonical for the most part, but there's some part that doesn't. What we want to happen is for the restraints to allow deviations where they're appropriate, but keep it canonical or similar when there is no experimental evidence. Now, this is a case where there's a side chain. So this is the reference side chain position, starting position, and ending position. And you can see it's maintained more or less this position up here, even though the target geometry from the reference structure was out. But because of the pull of the density and the non-harmonic nature of the Gemin-McClure restraints allow it to maintain its position in density, which is exactly what we wanted. So this is from the tutorial that Anna Casagnal and I wrote. So we have, again, using the CPF from Laurie Passmore. So we have a blob of density, a WD-40 domain. We do fitting a homolog, and then chop out this section and try to fit it, jiggle fit it first. That puts it over the density, but it's the wrong shape. So then we add in the homolog, add in the local distance restraints onto the homolog, and turn on refinement, and that ends up something like this. It moves side chains that are out of position like that allows it to refine into position. As I said, this is the homolog, and so there are places, for example here, where this particular loop is the loop of the homolog, not the structure that is involved in the reconstruction. There's another example here. Maya Topf and team wrote Flex-EM and wrote a tutorial for that, and Greg Pintilli and Wachu wrote a paper more recently that uses the same structure. The idea is that we're going to fit 4KI8 into EMDB 6422, and it involves some domain rotation. So it's a rotation translation. Fit doesn't do the job, as I'll show you here. So this is Coot's fitting. You can see that you wouldn't be able to rotate it or translate it to make it fit better, but it doesn't fit very well. This is also a Coot screenshot. Okay, so this is post-molecular placement, but pre-fitting. And so that is the fitting algorithm results there. Then this sort of operation takes about 30 seconds. Let's look at that from the other side of view. From the other side, you can see those helical pair of helices on the right there don't fit very well, but when we use Gemin-McClure restraints in the real space refinement in Coot, you can see that whole domain tipping and going into where we hoped it would go. Okay, so the above line is the density scores before and after fitting. You can see it's a rather better Fit. Coot isn't the only game in town when it comes to interactive refinement of cryo-EM reconstructions. There is another option that's Isolde, and it's based on Chimera X, or built into Chimera X. It's a plugin, I think that's the term, maybe not. You can get it from the toolshed. It involves Clipper, OpenMM. It's attractive and powerful, and Tristan's implementation of this and demonstrations inspired me to add what I call updating maps and proportional editing into Coot. So if you want to see a recent paper about that, is you can see the paper at the bottom of the screen, and Tristan also has some tutorials on YouTube. Okay, ligands. Moving on to ligands. We've built the protein, we've fitted it, now we're handling the details of the small molecules. So ACE drug is the generator of chemical restraints that we're using for our refinements. It does, obviously, as you can see there, ligands and polymers. And importantly, it's derived from the COD, which is the crystallography online database. That means it's unencumbered by intellectual property concerns that may concern me, if one were to use other well-known structural databases. Yeah, just leave it there. And unlike unlike previous attempts for creating dictionaries, and I had libcheck in mind, the atom types are exquisitely detailed, and they contain second order and third order information to accurately type the atoms. And I think there are many hundreds of thousands, I don't know exactly the details, of atoms, whereas there's only 200 atom types in the previous dictionary. And this was written up in 2017. Right, so second order neighborhood information. What do I mean? Well, let's compare two atoms here. First of all, this one. So this is called H1b. That's a name. What this means is it's a hydrogen connected to a carbon that's connected to a hydrogen, another hydrogen, and an oxygen. A hydrogen that's connected to a carbon that's connected to a hydrogen, and an oxygen. That's its type. It's about as simple as it can get. Here's a different example. This carbon here, and this says it's a carbon in three rings, two 5-membered rings and a 6-membered ring, and it's connected to 1, 2, 3, 4 atoms, 3 of which are carbons in various rings, and a hydrogen. So this carbon here connected to two. It's connected to a carbon in 2 5-membered rings, and this is a carbon in a 6-membered ring. So let's look at this here, and these are the carbons that it's discussing. And that hydrogen there is, of course, that hydrogen there. Right, and so you can see that the atom types are detailed. Trawling then the COD, the idea is to create a set of reference mean and standard deviations with outlier rejection, so that when you can take your particular molecule, I can identify those atom types and look up those atom types in the tables and determine what the expected bond length and standard deviation should be. Likewise for the angles. Now, a caveat for this, at least at the moment, that ACE drug doesn't do metals, but that's being worked on. Okay, so relatively new addition to ACE drug is the ability to determine links, and this was a difficult, time-consuming, multiple days of effort and understanding were needed to create dictionaries for links, and it was easy to make mistakes. And as I said, it requires exquisite care and understanding of the chemistry that you're trying to model. These days, with the new version of ACE drug, this, all we need to do is to tell it the bond order, and it can determine the bond order and the atoms that are involved in the interactions, and ASTRUG does the rest for us. And this was written up recently by Rob. This is a figure from the paper, and what we're looking at here is what ASTRUG is providing in that new link dictionary. It's providing new bond and angle restraints, new torsion restraints for these atoms here. Oh, this is pyridoxal L-phosphate binding to a lysine via a shift base. As I said, so torsions are the new is in green, and there are plane restraints that have been removed, and there's a plane restraint that's been added. Now, this figure was made in Coot, and this is using the programmable shaders that I'll mention in a minute. They're under user control, and I used user control to get that kind of blobby effect with transparency. Okay. Carbohydrates are going to be skipped over. How does it work in Cryo-EM? So this is a structure with which you may be familiar these days. Yes, it's the spike protein of SARS-CoV-2. It is slightly different, perhaps, to what you typically see. It's decorated with carbohydrate. And I did a grep of the recently published structures that have carbohydrates in, and this is the one I found. And there are issues. So if I turn on that's not great, but if I zoom in a bit, what I'm looking at here is the pore interactions. You may be familiar with mole probity dots. This is the Coot version of it. And you can see that there are spikes going on at the carbohydrate positions here. Here's one, here's carbohydrate, here's another one. This one is okay, but there are, as you can see, there are lots that are not. And here's a typical example of that. So there's a problem of the link. Now, if we use Coot's model building tool for carbohydrates, we can turn something like that, which is, there's an extra atom down here. I don't know whether you can see it. It's both blue and red because the oxygen hasn't been removed. And over here, there's a missing alpha-1,6 link. The missing link, as Rob would call it. In about a minute, that can be rebuilt using Coot's rebuilder. So there are friends of Coot that have been developed in Gribb's group, Servalcat and EMDA. The idea for, well, the Servalcat in particular, is something that I'm keen on because it allows us to do something that's turned out to be very useful in crystallography. That is to say, as we modify the model, update the difference maps. So this is what's going on here. It's based on GEMI, and it was recently published. So this doesn't exist in Coot yet. There's no interface to this, sadly, but there should be soon. And so what that means is as you do your model building operations in Coot, you can have the difference map updating as you do so, which gives, well, it's a pleasure to see. So here are some examples. No matter how many molecular dynamics trajectories you use, it isn't going to flip around that peptide, and looking at the difference map for a cryo-EM reconstruction turns out to, well, it would have been useful in that case. This is, you may have heard of the Cryo-EM challenge. This was a structure in the most recent challenge, 7770, beta-galactosidase. The experimental scientists who are doing this rather pushed, I would have said, the exposure, wouldn't you? And so everything that's charged has just disappeared from the reconstruction. Now, you could argue that the B factor is wrong or the scattering factors are wrong, but even if I push up the density, push down the density level, there's not much there. It would be interesting to do a frame-by-frame reconstruction of this in the style of Chris. Right. Okay, a few screenshots. there's no more intellectually um, challenging, perhaps, issues. So, as you may know, Chimera X has undergone a rewrite, or Chimera has been rewritten as Chimera X. It's still underway. There's not everything that Chimera does that Chimera X does. One of the things that they have changed is to use to a single panel view. And so Coot is moving along the same lines, that the whole of the application and its interaction should be in a single panel rather than these floaty dialogues that you can put underneath the wrong window and get lost. Right, so the sequence view, Ramachandran plots, geometry analysis are now part of the main application. If you care about Python, Coot is now available as a Python module. As well as the real space refinement, the lighting and representation have been improved. So this is what this is from the Chimera X documentation, and you can see what a great improvement using ambient occlusion and shadows provides to the interpretability of, well, in this case, this is a molecular surface, but it would also apply to reconstructions as well. It does, as you know, if you've seen any papers over the last 2 years. Right, so I wanted to bring some of that into Coot as well. So, the flat shading makes it difficult to perceive depth. Modern hardware allows post-processing of the image, frame buffer techniques, and that's what Coot is now using for more realistic lighting. And I've got a few screenshots of how that looks. So this is the old Coot, and it has a lack of depth. The only thing it has to confer comprehension of depth is depth fogging to blackness. That's okay when you're looking at side chains at high resolution, but most Cryo-EM reconstructors don't look like that today. They look more like that, and Coot is just terrible at. Coot is terrible at. Old Coot was terrible at displaying reconstructions, and for good reason, people use Chimera and Chimera X to do so. So this is Coot's new render engine, and already you can see that there's an improvement. Now you can see that the structure is made of atoms, but when we add the post-processing extra lighting effects, you can see hopefully that the configuration and juxtaposition of the components of this ribosome particle here are rather more readily apparent than they were in the naive lighting scheme. We can use that, oh blimey, pixels. We can use, for small molecules, protein-protein interactions, electron density maps. So this is how it used to be, old Coot, new Coot. We can have solid surfaces and ambient occlusion and this kind of clay effect. Hopefully that is going to improve the interpretability of maps. We can have transparent maps, which are a bit shiny. We can have depth queuing as well, and segmentation. We can have, what's it called? Fresnel effects on the electron density, which we can turn up a great deal to make the color black. It's still an interpretable electron density, even though it's black on black. And the reason why is because of the way the color plays on the surface of the electron density. This is electron density. So that you can see the shape of the map. And that was a surprise to me. A black on black effect works very nicely. The maps can be super sampled, multiple contour levels, and this is a map that you have seen before. You can take one map and contour it by one map, and color and contour using different maps. You can use build files to represent preferred orientation. You can use map masking, and this is again SARS-CoV-2, spike protein. You can use it for ribosomes, and that works rotating at 60 frames a second just fine. And I don't know whether you can see that. That's the old Coot versus new Coot. The one on the left-hand side, to my eye, is flat, and you can't really see that you're looking at a cavity between two subunits in the asymmetric unit there, whereas on the right-hand side, it's quite clear that there's a big gap between the two domains. To my eye, at least, I hope it is to you too. So as I mentioned, this is all done with improved lighting. And the way this is done is programmable shaders. So you have access to these programmable shaders, of which there are 50. They're written in GLSL, and you can change them. You can change them to however you like. They're overridable by the user. Okay, so this program was released last year to mixed reviews. It turns out a surprising number of people tried to use it on Macintosh, but the performance there was rather terrible. I'd been using GTK3, and the GTK developers were not interested in fixing that because they're using GTK4. CCP4 didn't want to distribute that version of Coot because it was too slow on Macintosh, just looked ugly, and the window resizing made things just uglier still. Okay, so the idea then was GTK4 was the solution. This is it turned out to be my lockdown project. The whole GUI needs to be rewritten again, and now this time there's no canvas, so there's no way of representing Ramachandran plots, no graphs, no ligand builder, no sequence view. Well, all right, I'll do it, but reluctantly. Unfortunately, global phasing, I was unable to secure funding by other means, let's just put it that way. Global phasing recognised the need for this project to be pursued and funded, thankfully, Jakub Smolski as a freelancer. And he's been very helpful. He teaches me how to use the widgets, which is great. It's all going on public. If you want to follow it, it's still not easy to compile. We can come back to that in a moment. But here it is in action, the new rebuilt interface, rebuilt from scratch. The GUI and the GUI's changed, but the graphics stayed much the same. So the tutorials were written for game developers. If you're going to be using OpenGL, then what else would you be doing other than writing a game? So so what I'm going to do is make Coot available on the Steam Deck. Not tomorrow, though, but at some stage. So what I wanted to be able to do is to provide a feedback in the way that you'd expect to feel from your typical experience of interactive 3D software, let's call it that. So one of the ways we can do that is spend some time in our 16.6 milliseconds to analyze the restraints. And they become interactive and representation in colors of, uses the color scheme to represent energy, or Coot's version of energy. Right, so there's a lot going on here. This is all dynamic and interactive. So as you pull around the structure, the Ramachandran plot changes. You can click on the Ramachandran plot to go to places in the structure. The probability of the rotamers is denoted by a color of the ball, and the probability, sorry, I got that wrong. The probability of the rotamers is denoted by the color of the spiky dodecahedron here, and the color of the ball represents the probability of the Ramachandran. The idea is that we want to turn the green, the stuff that's orange and yellow and red into green. So green is high probability and red is low probability. I know not everybody can see colors, and there are plans in the irons in the fire to make that more readily interpretable for those who can't tell the difference between red and green, which is quite a few high percentage of men. Right, and we can also analyze the structure as it's undergoing its refinement. And what this is, instead of writing information to the terminal, I'm writing information into the screen here. And of course, this is all done with the gaming techniques of textures and instancing. Because that's all I know how to write a game. So this is what you get in Coot. This is all interactive here. The pink bar under the residue that these are all interactions. And the pink bar under this interaction means it's the residue that you're looking at the center of the screen. So if you click on something with a pink bar, you take it here, and this is, if you could rotate the view, you can see this is the wrong rotamer. I just will flip the rotamer here, back grab rotamer, and it will flip into the right place, and, well, maybe we'll have a chance to do that in a demo, and the unhappy faces will disappear. We can use markup for ligands, so there's a recent change to the way that the markup works. It now uses a dictionary, which is very typically an ACE drug dictionary, and you can compare and contrast how your particular model compares to the specified geometry in the dictionary. And there are 3 methods by which your ligand is assessed. This is the quick ligand validation. So density correlation, geometry distortion, and ligand interactions with the protein. And so we've got the wrong one around here, so it doesn't fit well with the density. It is well refined though, at least internally, but it also is making poor interactions with the protein. So when we put it around the right way, all of those factors are improved. We can do surfaces with those interactions on them. Clashes, as we saw before, rather more clearly here than I was showing you in the spike protein. Okay, I'll skip over Moorhen. Moorhen is Coot on the web, moorhen.org. There is an interface that we can now use for using Coot with Blender. So there is some interest in the interface with molecular nodes from a week or 2 ago. This is something that Lucrezia and I have been working on. So we take the Coot libraries and then add an interface or a GUI in Blender speak, and then we have access to crystallographic functions or Cryo-EM functions where we can read in molecules, dictionaries, CIF files, restraints, MTZ files, and fetch things from the PDB, maps, and EMDB, and use that to construct our initial geometry, which we can then play around with the normal Blender tools. So this is me importing a dictionary from ACE drug and using a technique called inverted hull for the outlining. Subsurface scattering, ribbons, more ribbons. This is again CPF3EOJ. Gaussian surfaces where I change the material of one of the domains, DNA and RNA. Map masking and map sharpening and blurring, carbohydrates, electron density, and map masking again. This is a kinase inhibitor, checkpoint kinase inhibitor. Right, okay, I've talked about protein a lot. What about RNA? I don't have much experience with RNA. That's why I haven't really mentioned it. I don't know how one would go about modeling it. I should perhaps get some experience, but I don't have any at the moment. There is a tool called rCrane that Kevin Keating and Anna-Marie Pyle wrote some time ago, and that was added as a module into Coot. It hasn't been ported to the new GUI and needs some love, but it used to work, and you may find it useful. All right. So the user interface of Coot has been improved. Particularly the graphics, the ability to perceive depth, because we can now do more frames per second. The interactivity of the program has improved. You can get updates on my blog and on my YouTube, where there's a few tutorials. So for Cryo-EM summary, we now have, compared to how life used to be when I started working on improving the tools, well, and becoming a member of the team, I suppose, so improving the tools for Cryo-EM reconstructions. We now have a good deal of powerful tools for addressing these issues written by ourselves and others. So it turns out, it seems to me, that modeling and refinement isn't the bottleneck that it used to be. And improving the automation of the tools that we have and the issues of software freedom, which I am concerned myself, will be ongoing issues. What about problems with 4 angstrom? Don't know. What do we do about borderline cases? What do we do about cases where we want to encode our comprehension of molecular flexibility? How do we encode that or represent that in our results, in our models? That's still problems yet to be resolved. Thank you to my colleagues in the lab for useful discussions, and Lucrezia in particular for the blend Coot interactions. Bernard and Kevin have been long-term contributors, and Bernard looks after WinCoot so that I don't have to know anything about Windows, thankfully. I'll talk about Moorhen the next time. Thank you to Philo and Stuart and Martin there, and thank you for your attention.